you've probably forgotten. So hopefully it's like, I've reached the stage where when I read a novel that I've already read, I can't remember anything. So it's like reading a brand new novel all over again. So I hope, you'll find that talk just as interesting as when I reread a novel. So I'm going to talk to you about work that we've done on initiation. Actually, this work really started, we started with factors in the 90s, and then we started working on, the yeast system in around 2002. And it's just continued with many series of postdocs. And so what I'm going to tell you is really some of the more recent findings in this long process. And we still don't understand really the details of initiation. So what you're going to get is really just a little beginning, if you like. So this is just for those of you who don't think about the ribosome all the time. This is a small subunit which binds messenger RNA and presents the codons to tRNA. This is the P site tRNA that holds the nascent peptide, and this is the A site tRNA which brings in the new amino acid. Now, the problem is the ribosome has to start somewhere, and that process wasn't very well understood. And initially, when ribosomes were discovered, it was known that ribosomes had large RNA inside them. And the early idea was that, well, first of all, initially people didn't even know if DNA or RNA was the template. But when ribosomes were discovered, they thought that the ribosomal RNA was the template, and each ribosome carried its own message as its template. And so that changed with the discovery of mRNA, which was a short-lived RNA that was proved to contain the translating message. And so it became clear that the ribosome didn't carry its own template, but it was a universal translating machine that translated all mRNAs for all genes. And when mRNA was discovered, people still didn't have an idea of the process. They had this idea that maybe it just starts at the 5' end, keeps going until it reaches the 3' end, and then simply falls off. And that changed when termination codons were discovered. They were originally called nonsense codons, and people like Sidney Brenner discovered them. And they turned out to be termination codons which signaled the end of the coding sequence. Then, shortly afterwards, a special codon, which was AUG typically, but can be occasionally, later it was found you can also have things like CUG. But the point is you have a special codon which codes for methionine, which isn't at the 5' end. It's somewhere downstream. So there's an untranslated region, a 5' untranslated region. And similarly, there's a 3' untranslated region after the stop codon. And the tRNA that recognizes this start codon is different from the tRNA that codes for methionine within the coding sequence during the elongation process. So you have a special tRNA and you have a start codon. So how does the ribosome, there are AUGs all over the message, and there might even be AUGs elsewhere. How does the AUG that is the start codon recognized by the ribosome, and how does it know how to start? And that's basically what the problem of initiation is. So in bacteria, there are several factors. First of all, there's a small piece of ribosomal RNA in the small subunit that's complementary to a stretch of the mRNA just upstream of the AUG. What this complementarity does is it puts the small subunit in the vicinity of the AUG, but not precisely at the AUG. What you want is for the AUG to be positioned in the P site and the initiated tRNA to occupy that P site, so that the next codon will code for a sense amino acid, and it'll be brought in the normal way by EFTU. So in order to position it exactly at the AUG, you need several components. First of all, you need the initiator tRNA. That itself is often enough to position it right at the AUG. But in bacteria, there are 3 protein factors that facilitate this process, and they're called IF1, 2, and 3, and they add fidelity to the process, and they also, in the case of IF2, they facilitate binding of the large subunit. Now, I've just drawn these. These are not exactly to scale. And if you look at eukaryotes, though, the problem is much more complicated. Firstly, the mRNA itself has a very specialized structure typically. It has a 5' cap, and this is a particular 5' guanine linkage which occurs at the 5' end. Then you have there's no shine Delgarno, and you have an AUG, so you have an untranslated region. Then you have a stop codon, and typically eukaryotic mRNAs have a long poly-A tail of variable length. And instead of 3 factors, there are over a dozen factors. But the 3 bacterial factors all have counterparts in eukaryotes. For example, IF3 is the ortholog of EIF1, and similarly IF1 corresponds to EIF1A, and IF2 corresponds to EIF5B. So why do you need, what do these dozen or so factors do? So the way it works is you start off with an empty 40S, and initially a number of factors bind to this empty 40S. Typically EIF3, EIF1, and EIF1A are thought to bind first. And then to this complex, what binds is a ternary complex, which is EIF2, which is a factor that brings that initiator tRNA to the small subunit. And it's called ternary complex because it also has GTP, so it has 3 components. And then there's another factor called EIF5. Now these, when they bind, form what's called the 43S pre-initiation complex. Now while this is happening, there's another factor called the cap binding complex, and this is a factor made up of EIF4E, 4A, and 4G. Now EIF4E is the factor that actually binds the cap itself, and EIF4G is a large scaffolding protein that interacts with many different components of the system, and EIF4A is an RNA helicase. So this cap binding complex, which is confusingly called EIF4F, which seems like it would be another factor, but it's actually these 3 factors. This cap binding complex binds to the 5' end with the 4E directly binding to the cap. Now at the same time, there's another protein called poly-A binding protein. This protein binds the poly-A tail at one end, but it also has a binding affinity for EIF4G. So the result is that you can get a topological closure of the mRNA so that the 5' and 3' ends are brought together by this interaction via PABP. Now the role of this complex of cap binding complex is to recruit the 43S to the mRNA. And when that happens, of course the 48S it's called the 48S complex. And then when that happens, it still has to find the AUG. So what it has to do is scan along the mRNA, and this is where the helicase activity of 4A is thought to be important. And when it finds the AUG, a number of conformational changes occur, and eventually the large subunit joins, and then you can start the elongation cycle, and eventually you go until you reach termination and recycling, and the whole process has to start over again. So you can see that there are a number of factors. Some of the factors like EIF3 have 13 subunits in humans and are about as big as the bacterial small subunit. So why is it that eukaryotes have such complicated a process of initiation, whereas bacteria can do it with just 3 factors and initiate a tRNA. So there are a number of reasons for that. The first thing you can think about is that in bacteria, there's no separate compartment. So when you have transcription, the mRNA comes out, is extruded by the RNA polymerase, and even before it's fully synthesized, ribosomes already start translating on it. If you've looked at your undergraduate textbooks, you may see the classic Christmas tree figures that were determined by Oscar Miller, showing DNA and then mRNA coming out like branches of the Christmas tree, and you can see little dots which correspond to ribosomes that are attached to it. Now, in eukaryotes, that's not true. You have a complicated process where transcription occurs in the nucleus, then it has to be spliced, then it has to be processed and modified in various ways, and then it has to be exported. And only then does translation occur in the cytoplasm. And this allows an opportunity to really control gene expression at a second level, which is at the translational level. And so in eukaryotes, it turns out translational regulation is almost as important a component of gene expression, of regulation of gene expression, as transcription is. For a long time, it was thought transcription was all of the regulation, but now we know that's not true. And I'll give you, so I basically told you this, and so you need more translational regulation in eukaryotes. For example, you have development, formation of memory, stress, infection. All of these involve translational regulation, and it's a cause of disease and aging. And before I plunge into our actual work, I just want to give you a couple of examples of how important translational regulation is. So this is the GTP exchange cycle. So what happens is the ternary complex, when it binds as a complex with GTP, EIF2 with GTP. But when it takes part in initiation, it hydrolyzes GTP. So you're left with EIF2 in the GDP form. This binds to EIF2B. And what this does then is, which acts as an exchange factor for GTP to GDP to GTP, and that then allows the binding of initiated tRNA and then formation of a new ternary complex which can take part in initiation again. But there are a number of kinases which will phosphorylate EIF2, and when that happens, then the phosphorylated EIF2 in the GDP form binds to EIF2B and then cannot dissociate. And so it can't be released. And so it's essentially sequestered. And effectively, there's no, the cell runs out of GTP form of EIF2, the ternary complex. And so it essentially results in an inhibition of translation. And these kinases are activated in response to a number of things. For example, one of the first kinases was discovered by Alan Hinnebusch at NIH, who showed that GCN2 acts in yeast during amino acid starvation. And another, this one, HRI, acts during the lack of heme. And then you have PERK, which acts during viral infection, and then another kinase acts during sensing unfolded proteins in the endoplasmic reticulum. And so you can see this is called the integrated stress response, because you can get stress or aberrant situations due to a variety of reasons, but they all act a common point. They all phosphorylate EIF2, and that's how they shut down global protein synthesis. But it's also it's interesting that they don't just shut down global protein synthesis or inhibit it to a low level, but they also, by some clever pathways which avoid the usual routes of initiation, they turn on special genes including genes for amino acid biosynthesis. So you can imagine when a cell is starving, you don't want to keep trying to make proteins, but you do want to make proteins that actually make amino acids so that you rescue yourself. And this has implications in both diseases and in aging. Similarly, you may have heard of the TOR pathway, which is a nutrient sensing pathway which globally keeps protein synthesis turned on. It's essential for growth, and it's a hugely important metabolic pathway. And it turns out TOR is a kinase, and one of the ways it acts, it has many, the TOR pathway has multiple effects, but one of the things it does is the kinase phosphorylates a protein called 4E binding protein. In the phosphorylated form, this 4E binding protein is unable to bind 4E, which is the cap binding protein. And so as long as TOR is active, which is under normal conditions, translation is not inhibited. And so protein synthesis can carry on as normal. But if you inactivate the TOR pathway, if you inactivate the kinase, and you can do this by either nutrient deprivation or you can do it by drugs like rapamycin, then what happens is that the 4EBP is no longer phosphorylated, and it can bind 4E, and when it binds 4E, 4E then is no longer available for initiation, and it essentially reduces global protein synthesis. So I've just shown you two examples of two very different but extremely important metabolic pathways which are regulated by controlling translational initiation. So we wanted to understand how the whole thing gets started, and we wanted to understand in particular how the cap binding complex recruits the 48S subunit and starts scanning. So if you look at the 4F and look at what was known when we started, there was a crystal structure of EI4E, which is the cap binding protein with a cap analog. There was also a crystal structure of this helicase, EI4A, with a piece of RNA in both the closed and open form. And then the EI4G was actually mapped. In other words, it's a long scaffolding protein, about 1,600 amino acids in humans. And it has poly-A binding domain, a 4E binding domain, two 4A binding domains, a domain that interacts with EI3, etc. And this particular part is considered the very core of the EI4F, because they found out that even if you delete this, you can still get initiation via EI4F. So what are some of the questions that we might be able to address with this 48S complex? Well, one is, there was a question of how the 43S is recruited to the mRNA. Well, the obvious way it would be recruited is if you have 4F binding at the 5' end, then the only way it could be recruited is downstream of it. But since this part no longer has a free end, it would imply that the mRNA, there's something weird going on with this. Anyway, it would imply that the mRNA is slotted laterally into the mRNA binding channel of the 43S, of the ribosome, so small subunit. The other model that was proposed by some people was a so-called threading model. In this model, the 48S, the EI4F, the cap binding complex, would actually bind at the site where the mRNA would enter the small ribosomal subunit, at the entry site. And then somehow 4E would dissociate, and because of the helicase activity, it would drive the mRNA and thread it through the small ribosomal subunit, through its channel. Now, why would you, this would be a slightly complicated thing where you would have to bind, dissociate, and then thread through. Why would you propose a model like that? Well, the reason to propose that is because there are mRNAs which have a very short UTR, even just a few nucleotides. And if you had to slot it in here, then the 5' end would be quite far from the P site. And so it would be incompatible with mRNAs with very short UTRs, where the AUG would be over here somewhere, and that would just not fit with this sort of model. So people in the field had proposed that. So that was one question. The other question is, how does the 40S actually scan along the mRNA? And the third is, what happens when it reaches the start codon? So we don't have all the answers at all, but I can just say that we've made a beginning, and you can see how far we've gotten. So we had been working on the yeast system for a long time, starting first with just the 40S with a couple of factors, and then with people like Tanvir and his colleague Jose Yasser looking at bigger complexes of the 43S with mRNA. And for a long time we had tried to get the structure of the entire 48S complex, which includes the cap binding complex. And it never seemed to work, even though it seemed to be stable in solution. When they did EM, they could never visualize even any component of the 4F complex. And I can tell you in hindsight why we think that is. But in the meantime, Chris Fraser, who was at the University of California in Davis, and his longtime colleague Masa Sokabe approached me and asked if I would be interested in doing work on the mammalian system. And of course, you know, we thought that would be a great idea to complement studies on yeast. And the work was really spearheaded by this guy, Jay Querido, who's now a faculty member at the University of Michigan at Ann Arbor. And he was joined in this effort by a number of people, including Irene Diaz, who was also another postdoc, Yulia Gordienko, who's a long-standing research associate of mine, and Sebastian Kratz, who was another postdoc who helped Jay in the first part of the story with a number of computational aspects. So the first thing they did, the advantage of collaborating with Chris Fraser is Chris had purified all of the human initiation factors. And this is, those of you who are under 30 probably don't understand what purification means, because you're used to cloning a gene with some sort of tag, and then you put it on an affinity column and you elute it, And that's not what, classical enzymology is. In classical enzymology, you lyse the cells, there's no tag. You have to figure out how to purify it by a whole series of columns, by ammonium sulfate precipitation, and so on. And so that's the kind of thing he had to do with 20 liters of HeLa cells. And so that was the advantage. And the advantage was that with purified factors, you could reconstitute a 48S complex from scratch. And so then we had to ask, well, is this complex actually active? And so to do that, Masa Sokabe used an assay called the Raleigh assay. And since I'm going to be describing it later, I want to tell you how it works. So the way it works is you have an mRNA and you have an AUG, for example, at position 50 and another at position 60. And then you tag this mRNA with a 3' fluorescent label. Now, typically what will happen is your initiation complex will scan along the mRNA until it reaches the first start codon, and then it'll stop there, and because that's the initiation codon, and it won't go any further because it will require the large subunit to join in order to be able to start translating. But if you don't we don't put the large subunit there, so we would expect all of the initiation complexes to reach the start codon and just stop there. Now, what you can do is use an enzyme called Raleigh, which is an endonuclease. And since the A site is empty, this is a nuclease that cleaves mRNA in the A site of ribosomes. It won't cleave naked mRNA. It's very specific for cleaving mRNA in the ribosome in an empty A site. And so what would happen is, if you cleave a RelE, you would get cleavage right here or right here, depending on which AUG the initiation complex installed. So if you did that, what you see is following. So this is just, the uncleaved mRNA and degradation product. But here you can see that some fraction of the complexes have reached the position AUG at position 50 and stopped there. And there's a little bit of read-through. So some of them have read past the AUG because the stop recognition is not perfect in the system, and then gone to the next AUG and stopped there. If you had ATP gamma S, which is an ATP analog, supposedly non-hydrolyzable, but actually it's slowly hydrolyzable, then we see pretty much the same thing, because on the scale of this experiment, it acts pretty much like ATP. But if you use a much more non-hydrolyzable analog, like AMP, PNP, you can see that you've really reduced the number of ribosomes that have reached the start codon. And you can quantitate this. So for example, this is with ATP and this is with ATP gamma S. And this is if you leave out the initiation factors. So what you can see is this is both ATP dependent and factor dependent. And so we felt that this is a physiologically active system, and therefore we were ready to do the structure on it. And here's the structure. I'm just going to show you the structure just once around. And the important thing is this was about a 3 angstrom structure for the most part. So it really had the best resolved 43S complex to date, regardless of whether it was mammalian or yeast. And in fact, you could trace all of the 13 subunits of EIF3 in the structure. And so it resulted in a very complete 43S complex. But for us, the really interesting thing is that there were additional blobs of density that we thought corresponded to EIF4A and 4G. And strikingly, they were not at the entry site, which would be over here, but they were at the site where mRNA would come out at the exit site and then presumably bind 4A. But the mRNA here was too disordered, so we couldn't actually see the mRNA, partly because the resolution was really not at all good. Now, we did not have a start codon in the mRNA we used. And so we presumably, if we had a complex, it would presumably be in something like the scanning conformation. And so earlier work on yeast had shown that the 40S can occupy 2 states called a P out and a P in state. So when it hasn't reached the start codon, or if you have a non-cognate start codon, then the head of the 40S is in a more open state, and the tRNA is somewhat further away from the start codon. So you're not making the proper base pairs. But when it reaches the start codon, there's a global conformational change. And what that means is that this head closes in and the tRNA is buried more deeply into the P site to form what's called the P in state. Now, if you look at the structure of this complex that I just told you about, the tRNA is somewhere in between that of an open and a closed state. And so we think it might represent the scanning conformation. So remember, during scanning, the tRNA has to constantly sense whether it has reached the start codon or not. Otherwise, how does the system know that the start codon has been reached? And if it's constantly base paired, then of course it can't slip. It'll be energetically difficult to slip. On the other hand, if you move it too far away, then sampling becomes energetically more expensive. And so the idea is to, if you have an intermediate state, then you can quickly sample as the whole system moves along the mRNA to scan. Now, if you look at the location of EI4F, we can only see really we can see all three components, we think, but we can only see parts of them. So you can only see the first heat domain, which is the 4A binding domain of EI4G, and we can see what we think is EI4A, but the density was very poor. But in any case, when we interpreted this, what we found is that this 4F essentially makes interactions with EI3 and appears not to make interactions with anything else. But the more worrying thing is it makes interactions with subunits of EI3 that are not present in yeast. Now this is very peculiar because both yeast and mammals have EI4F and presumably, and they have EI3, so presumably they ought to work in the same way. So why would you have this 4F binding subunits that don't exist in yeast? You know, it seemed a little odd. So we went ahead and did some mass spec cross-linking mass spec experiments to try and verify that these contacts actually existed in solution, and satisfied ourselves that it was probably true. Now, another blob of density that we saw here we think might be EI4E. So you could imagine the mRNA coming here and then ending up in 4E, which actually has the 5' cap. But of course, we can't see the mRNA, and it's a blob which has the right shape and size to fit 4E. And of course, we had 4E in our complex, so we think it's probably 4E. Now, what this structure does is it tells us something. That is, we can see the mRNA inside the 40S, but we can't see the mRNA once it's left the 40S, and certainly we don't see it where 4A and 4G is, because the whole system is highly disordered there. But we can estimate what this length is, and if we estimate what the length is from here to about here, then it looks like it's about 30 to 40 nucleotides. And remember, this is not 4E, so in fact for 4E it might even be a little bit more, okay? So what this means is that in order to initiate, your AUG cannot be any closer. I mean, this is even without scanning. So it can't be any closer than about 30 to 40 nucleotides. And so there was some evidence for this because Marilyn Kozak, who studied initiation, showed that if you have mRNAs that have shorter UTRs, for example, less than 32 nucleotides, you get what's called leaky scanning, which is that the system doesn't initiate efficiently on the first AUG, but keeps going until it encounters the next AUG and then initiates on that. So we put all this together and submitted it, but of course, reviewer 3 was very unhappy because, you know, it's a low resolution structure, at least for the 4F part, and we're sort of hand-waving our way through it all. And also, it puts the 4A on the wrong end of the ribosome. You know, 4A is a helicase meant to unwind mRNA. It's actually behind the 40S. So Masa Sokabe decided then to do a very clever series of experiments, which is he took different mRNAs. All of them had an AUG at position 50, but then each mRNA had a second AUG at positions 10, 19, 30, 40, and 60. And the idea is that if these AUGs are not accessible, you shouldn't see initiation on them, and you should only start seeing initiation once the relevant AUG is 30 to 40 or more nucleotides away. And so this was his result. So when you have 50 and 60 for the AUGs, it primarily initiates on the first AUG it encounters, which is position 50. But if you have 40 and 50, you get some initiation at 40, but it still mostly initiates at position 50. And if you go to 30, 19, or 10, you see almost no initiation at those earlier AUG codons. And this shows that those earlier AUGs are not accessible to the system. Okay, so the minimum AUG you require is about somewhere between 40 and 50 nucleotides. And so is this model compatible? Well, we think it's, so the idea is that you have the EI4F, and the 43S is recruited downstream of it. And so it's not the threading model, it's the slotting model. And you then, it can then scan along, but this means that that you have to have a minimum distance between the AUG and the 5' end. And so this model is compatible with the typical 5' UTR of mRNA in humans, and even in yeast. And even in yeast, the median is over 50. And in humans, the median is supposed to be about 200 nucleotides. So it's perfectly compatible with most natural mRNAs. Now, there are short UTRs, mRNAs with very short UTRs, and we think that they probably initiate by a completely different mechanism. And there's an analog for that in bacteria. And bacteria have leaderless mRNAs. Those don't have a Shine-Dalgarno. They initiate in a somewhat different way from the typical process. Now, the 4A here is upstream, and so how would the helicase activity work? So we suggested that maybe it acts by pulling the mRNA through the 40S, and it may be that the 40S itself, it may be that the 40S itself is a helicase. And there's some evidence that the 40S, the small subunit, can actually be a helicase. And the other possibility is that the helicase acts as a pall to prevent backsliding. That is to say, it keeps going along with the 40S but doesn't allow it to go back. But there is also another possibility, which is, could there be a second 4A in the system? Why would you think there's a second 4A? Because this complex, when we isolate it, seems to have a 4A, so where would the other 4A be? Well, one issue is that EI4A is present in considerable excess over the other factors in the cell, and it's also known to have additional roles in melting RNA secondary structure outside of initiation. For example, it's thought to dissolve RNA stress granules. And there's a lot of biochemistry, including some by my yeast collaborators like John Lorsch, who suggested it's near the entry site, and they weren't very happy when we published this paper saying that 4F is on the other side of the 40S. So the question is, could there be a second EI4A binding site, which is at the entry site, which would also make sense for a helicase, because this is where the mRNA would be entering the 40S and needs to be unwound by a helicase. So in order to do a second experiment, Jay had a couple of goals in mind. One was to try and get a better resolution structure of the 4F, because it was really very blobby. We couldn't even see any secondary structure elements. So what he did was use these polypurine tracts, which in combination with a drug called ROCK-A essentially prevents EI4A from scanning, essentially locks it in place. And so we thought if we did that, then maybe we would have a better structure. And we weren't actually thinking about a second 4A when we did this experiment, to be honest. And this particular mRNA has an AUG here as part of its sequence. And the other thing to remember about this mRNA is it's quite a bit longer than the original mRNA, and that's partly because we introduced these polypurine tracts. So when Jay and his colleagues did the experiment, what they found, to my considerable relief, was that EI4F, that is the 4A and the 4G domain, was found in almost exactly the same site where we had seen it before. And the resolution was now better, and we were more confident that it was correct. But in addition, they saw density at the other end, at the exits, at the entrance site of the mRNA, and part of the density could be very well fit with the helicase, EI4A. And so the second site is at the entry site, but the other interesting thing is we could see it with or without ROCK A. And in fact, it wasn't clamped at the polypurine tract. It essentially scanned all the way until it reached the AUG codon and stopped there. So this was almost like a start codon recognition complex, similar to what we had done before with yeast. So why did we not see the 4A at the entry site before? Well, it turns out that the previous mRNA we had used was too short. And so the mRNA was, because so much of the mRNA is used up by 4F at the 5' end, there was not enough mRNA to come out at the 3' end, out of the 40S subunit. And so there was no real binding affinity for 4A at the entry site. Now, we could only know that in hindsight, because when you do the biochemistry, what you find is you find you're forming a very stable 48S complex, which has all of the factors, including 4A, 4E, and 4G. So there was no reason for us to suspect that this complex was anything other than a complete complex, because nobody in the field had expected that there would be a 4A that would act independently of the rest of the cap binding complex, which is 4F. So 4A was only thought to be part of the cap binding complex. There was no notion that it would act within initiation as part of the ribosome as a separate entity. Now, the other thing you could ask is, you remember 4G had two 4A binding sites. So could it be that this 4G is such a large protein, it's coming around, and therefore it's binding, this is bound to the second 4A binding site in 4G. We rule that out because you can take a truncated EI4G that lacks the second binding site, and you still see this second 4A. And so what this means is we have two 4As. One is part of 4F, which is recruiting the complex to the 5' end, and the other is a 4A helicase essentially by itself at the entry site, at the 3' end, where it's in a better position to actually play the role of a helicase. And finally, there's no room for 4G here, so it's not as if you can have a 4F binding at the entry site. Now, it turns out that we can, at low resolution, we can actually see much of the mRNA density all the way from the 5' end to the 3' end. And finally, there's some density that's extra density next to 4A at the entry site, and we suspected it was EI4B because this is a factor that enhances the ability of 4A to act as a helicase. And when you assemble the complex without 4B, then that density essentially gets disrupted. So we think that this is essentially represents EI4B, and that would make sense because EI4B would then be binding with EI4A and facilitating its helicase activity. Now, if you look at the 4F complex itself, the resolution is better. So, for example, you can see tubes for helices, and so we're much more confident now of our assignment of these factors. And the interesting thing is that they now make interactions with mRNA, not only with elements of EIF3, but also with elements of the ribosome that weren't seen before in our previous structure because the resolution wasn't good enough. And what you see is that this interface between EI4A and ES7 contains the site of ubiquitinylation of ES7. And when ES7 is ubiquitinated, then it inhibits initiation. And you can see how this would clash and you would inhibit initiation. So it makes sense in terms of a lot of biochemical data, but it also makes sense in terms of our own history, which is, for years we could never isolate a stable 48S complex in yeast. And that's because you can see that although yeast makes contacts, it makes far fewer contacts than the mammalian system. And that's why when you use the mammalian system, we were able to see, at least at somewhat lower resolution, parts of EI4F. and it was much harder to do so with yeast. So our conclusion is that there are at least two roles for EIF4A in initiation. One is as part of EIF4F in recruiting. It might be pulling the mRNA and possibly acting as a pol. The other is at the entry site, where along with 4B, it acts as a helicase, and it might be unwinding downstream mRNA secondary structure as it goes into the ribosome. And 4B may be a part of this. And in this structure, where it has scanned all the way to the AUG, we don't see EIF4E, which suggests that either it's dissociated or it's disordered enough that we don't see it. So what happens when the start codon is reached? Well, we can now see it in the mammalian system, but this is all work that was done earlier in yeast by Tanvir and Jose Yasser and their colleagues, along with John Lorsch and Alan Hinnebusch and a number of their colleagues. And remember, what I showed you was that you can have, in the absence of a start codon, you have essentially an open conformation. And when the start codon is reached, then you get a global domain closure and a burying of the tRNA deep into the P site, which is called the P-in state of tRNA. And this is just to show you what that looks like. It involves really large-scale domain movements, including lots of proteins and factors changing conformation in the process. Now, one thing that we had seen, again, first in yeast, but we now also see it in mammals, is that you can trap in a non-cognate tRNA using a mutant, you can trap EIF1 as part of the initiation complex. But normally, you wouldn't be able to, because the normal position of EIF1 would clash with the tRNA when it is being buried into the P site and recognizing the start codon. And so when you typically have the start codon recognition state, EIF1 dissociates. And it's only by using this clever trick that they're able to see EIF1 in the initiation complex. So what happens when you actually have a true initiation complex at the start codon? Well, what happens is that EIF1 dissociates, and it's then replaced by the N-terminal domain of EIF5, and that stabilizes the start codon recognition complex. And this sets forth a series of changes which we don't fully understand, where a number of factors have to dissociate, and then EIF5B has to join, and that then facilitates joining of the large subunit. And then eventually that dissociates and elongation starts. But you can see we've sort of mapped some of the early changes that happen when start codon is recognized. And we have similar changes in the mammalian system. So for example, we can see tRNA in the P-in state, whereas previously we had a scanning conformation, and the same EIF1 replaced by EIF5 NTD is observed. And so it suggests that the start codon process is fundamentally conserved in yeast and in mammals. So this is just a movie to show you the process. So here's the EIF4F, and this is the mRNA with the cap shown in light, and which binds EIF4E. And then this is the 43S complex, which is essentially has all of those initiation factors. And that's then recruited where the mRNA, as you can see, slots into the mRNA binding channel in the 40S subunit. And then scanning has to start. But to do that, you get the second 4A binding with 4B. And this then keeps hydrolyzing ATP and essentially scans along the mRNA. And eventually, the start codon reaches the P site. At that point, EIF1 leaves, and the N-terminal domain of EIF5 then replaces it into the initiation codon. And I started off by telling you about bacteria and how bacteria have 3 factors, and each of those 3 factors has a counterpart among the many eukaryotic factors. And this again is very much earlier work from 2016 by Tanvir and Jose and Brian Wimberly and their colleagues. And it shows some very similar features. For example, the movement of a head to close in and movement of tRNA into the P site. And this is just to show you what that looks like. So you can see that again there's a global movement and a movement of tRNA into the P site, and then this is the IF2 joining. And so even though you have over 1 billion years of separation, there is a highly conserved core on start codon recognition, where you have a global domain closure and a burial of the tRNA into the P site. So as you can see, we're nowhere near understanding initiation, but we've gone from sort of not knowing very much at all about what the process looks like to having some idea of some of the things that have to happen during initiation. So thank you very much.